I think an important message is that the value of your contribution to lab meeting is not in the quality of your performance, but it's in the content of your performance. It's what you bring and the questions you ask. And so you don't have to be polished. You don't have to have a final favorable outcome that you communicate. It's what you bring to the process, the questions you ask. That is your most important contribution.
iPSC are taken from patient samples, and we reprogram things like blood cells or skin cells so that we have a patient-specific stem cell that can then differentiate into theoretically any cell in the human body. And so then we have to prove that our stem cell lines are in fact pluripotent. And so we do things like this called immunohistochemistry for different pluripotency markers. For example, OCT4, NANOG is another pluripotency factor, SOX2, TRA181, TRA160. And you can see on the top here is the stain for the actual protein, and on the bottom is the isotype control, and those are all negative. The blue is staining the nuclei of the cells. And then another way to show that your stem cell line is pluripotent is to form what is called a teratoma. So what you do is you take your cells and you transplant them into an immunocompromised mouse, and they grow these tumors. And if your stem cell is pluripotent, it should be able to differentiate into cell types from all three germ layers that are formed in the early stages of development.
Well, no. Somebody working in Jochim's lab was trying to grow MDS cells in immune-compromised mice, and they wouldn't grow unless they co-injected them with 27A. So 27A provides them something and gives them a little boost. But 27A was made with a p53 knockout. I mean, that's how we made immortalized 27A. We took out p53, right? So I'm wondering if that's if a primary stroma that's not been immortalized would do the same thing. Something we may want to look at.
Right.
Because, just by chance, that's how those cells were made. They were immortalized with p53 knockout, HPV E67. I don't know if that means anything or not, but...
So this is sort of a bite the hand that feeds you phenomenon here. They need HS27, but they try to kill them.
Well...
I'm not sure they try and kill them because
maybe they just arrest their growth.
Yeah, that's what they do, because the cell number didn't decrease.
It didn't go down.
It just didn't increase.
Well, so we know from the work that Anisha and Louise have done that the stromal cells on plastic that will grow confluent and overrun the culture will not do it in a three-dimensional collagen matrix. And they won't do it here as well. So there's some sort of feedback that we need to know about. Okay?
So do you only see the growth arrest when they come into contact with the cells, or is it the whole thing?
We haven't really tested that. I mean, we basically just tossed them both into the same well and saw what happened.
What about conditioned media?
Haven't tried conditioned media.
Try conditioned media. See if that does it as well.
Well, it's a chance for us all to know what the next person's doing. And you can put your input in if you have ideas about how to redirect them or tell them how to fix their problems, or come across something where there's blocked and can't think of what to do next.
Like Jessie could be trying to do something that Gretchen did 20 years ago. She could just tell them how to do it.
And everybody's got different types of expertise. So GK and Mike are really experts in molecular biology, so anytime we have problems, they sometimes bring up things in lab meetings that we don't even know about or think about.
You have to be able to raise criticisms in a non-threatening way so that the person doing the work can embrace the criticism and adjust things accordingly. Otherwise, they can go down a path that's completely wrong. And there'd be nobody to pull them up short and say, wait a minute, think about this. So, for example, we were using an antibody to detect a certain kind of cell that was important to us to detect, so we could isolate it and study it. And it became apparent to one of the members of the lab that this antibody was not seeing what we thought it was seeing. And that was a hard message to accept, because we had invested considerable time and effort using that antibody for that purpose. But they showed unequivocal data that the antibody was not seeing what we thought. And so we just had to stop that series of experiments and step back. And that was important. And that happens all the time.